Okay, so I thought I would start off this unit with something that we can all relate to a little bit. And so I have a series of photos here that is displaying a process that has happened to all of us, I'm sure. And I want you to sort of think of how this is possible, or how our bodies make this process possible, because it happens all the time. So the first thing I want you to think of is that we have a cut here. And over time, so this is a series of photos of what's happening to this cut over time in your body. And for this to happen, our body needs to be able to regenerate the cells that make up our body in the first place. So another example, in a different type of organism, a snake can shed its skin. So if we think about how often a snake sheds its skin, this snake needs to be able to regenerate and recreate the cells that it's shedding. So if we go back to what you learned in grade 10, in grade 10 biology, we touched a little bit on a process called mitosis, and actually grade 11 biology as well. We're not going to go into detail with this, but I do want to talk about it just because it's important in the entire process of the unit we're about to talk about. So remember how I said in grade 11 you learn sort of on a larger scale of things that are happening, and then when we get to grade 12, we're going to zero in inside of the cell. So if you can recall, we went through the phases of mitosis. So mitosis is a process that happens in every organism's body that helps to regenerate cells, and in this case body cells. So this was the process where a cell grows, develops, and toward the end, in order for us to take one cell and make two cells out of it, we have to duplicate the DNA that's inside of it. So that was one process that happened before mitosis began. And then we would go through the process of mitosis, which I'm not going to go through in detail, and you do not need to be able to repeat this or know this for this course, but just on a larger scale, what's happening here. We just made two new cells from one cell to begin with. And the important thing I want to zero in on here is what's happening to the DNA. So remember, the DNA is really important, that every cell and every new cell has a complete copy of it. So this is really the important part, that we need to get our DNA copied before we can actually divide this into two new cells. So by now you might be asking yourself, okay, well, why are we taking the time to go through this? In grade 10 and 11, you may have spent a few days analyzing every single phase and look at in detail what's going on. Just to give you an idea of where we're headed in this unit, this entire unit is going to be spent looking at interphase in detail. So we told you in grade 10 and 11 that DNA makes copies of itself, but we never told you how it does. So this whole unit is actually going to focus in on how the DNA actually makes a copy of itself, and it's a pretty complex process. So that's why we can spend an entire unit on one phase of mitosis. Okay, so let's first start off by recapping what DNA is. In our first unit, when we talked just about the nutrients and what they're made up of, it fell under the category of nucleic acids. And remember, in that category, we dealt with two different types of nucleic acids, and we're going to revisit both of those. So we'll start with the first one being DNA. So the long name for DNA is deoxy. Remember, this is an important part. Ribonucleic acid. So some nucleic acids had an extra oxygen group on that ribose sugar. So DNA, deoxy, means it's without that extra oxygen, so it has one less oxygen. Now, what does DNA do? It is the chemical message that regulates life by carrying all of the directions for growth, repair, and replacements of cells and their parts. So you can imagine how important that molecule is to be intact and properly copied for every single cell in your body to have that same set of genes or copy of DNA. So it's found in every single nucleus of every single cell in the body, which is trillions of cells. It's packed tightly in a cell in the form of what we call chromatin. So chromatin is just the term that's given to the way DNA is packaged inside the cell, and I'm going to focus in on a picture in a minute. When it is unwound and stretched out, it looks like a ladder. So it's been twisted, and this is where it gets its name, a double helix. It's like a ladder, a flexible ladder that's been twisted into a spiral. So it's got the rungs of the ladder, it's got the sides, and then they've taken that ladder and they've twisted it. And that's what sort of makes up a DNA when we get down to the bare bones of it. In order to fit this DNA in our cell, this is what happens. It winds itself into the double helix, and then it crunches itself and wraps around itself into what we call a chromosome. Those chromosomes are what you guys dealt with in the phases of mitosis. Remember, we talked about, well, we didn't talk about in detail how they're packaged and what they're actually made up of. They're found inside the nucleus of every single cell. And so every single cell will have an identical copy of all of the DNA that a human body would, or any organism's body will contain. Okay, so back to what we briefly touched on in the first unit. We want to go back and just have another look at its structure. So now that we're familiar with some of these terms, we can now come back and analyze what DNA is made up of. So on the previous page, I just snapped a picture of this. This was just a small end of what we looked at on the previous page, but I can relate back to it so you guys can kind of get the big picture of this. So the DNA structure, these are two terms we should be familiar with. Something called a nucleoside, and then something called a nucleotide. Okay, so a nucleoside, these are just terms used to describe small subsections or units of DNA itself. So it's called a nucleoside when you just have your sugar and your nitrogenous base. So if we look at that chemical structure, this would be just a ribose sugar, and it would be attached to one of the four bases that we talked about, which I'm going to come back to as well. We call this, if it's just these two structures, a nucleoside. When we have this sugar, the nitrogen base, and at least one phosphate, it now becomes nucleotide. So really the only difference here is the S changes to a T, and those are the difference between those two terms. Nucleoside has the sugar in the base. Nucleotide, we have at least one phosphate. So these three things is what makes up the backbone of DNA. And that's why I brought this picture back in here. Oops. Where's my handy dandy undo button? There we go. Okay. If I bring this down and we compare the actual chemical makeup to what's happening here. Remember I said DNA is kind of like a ladder. Well, you've got the sides of your ladder, okay, is what makes up, so your sugar, let's see here. Okay, so your phosphates are these small little groups here, the small little circles, and your sugars are the bigger groups right beside it. And your nitrogenous bases, or nitrogen bases, which there's 4 to choose from, are all of the rungs that come across the ladder. Okay, so that's where we connect our chemical makeup to the actual structure of DNA. And remember that the pentose sugar is called deoxyribose because it has one less oxygen. And just to give you one extra piece of information as well, this is, remember, os means sugar, and pent, when you see pent, it usually means 5, and that tells you that there's a 5 carbon sugar. So 1, 2, 3, 4, and 5. Those make up your ribose, and ribose sugar has 5 carbons, so they call it in general a pentose sugar. Okay, so that's where the structure comes up. So your phosphates and sugars are on the sides of your ladder. Your nitrogenous bases come together that make up your rungs. Now let's review what those nitrogenous bases are. There are 4 nitrogen bases that exist in DNA. Do you need to remember or memorize what their chemical makeup is? No, you do not. You do not have to be able to draw these for me. But what you should be able to do is you should be able to recognize A, that they are a nitrogenous base, and B, what category they fall into. And I'll show you the 2 different categories we can have. Okay, so right off the bat, just looking at the 4 that we have to choose from that are found in DNA, you can notice that 2 of them have 2 double rings. So adenine and guanine have 2 nitrogen rings, cytosine and thymine have 1. And that's how you're going to distinguish the 2 categories. So here's the 2 categories that you can fall into. Adenine and guanine, which are both double ring structures, they're called purines. So this is important term to know. Purines is an important term to know. They make up double nitrogenous rings, and adenine and guanine are your examples. And the other category is pyrimidines. That's the two remaining, cytosine and thymine. They're single ring structures. Now, if you recall from the first unit that we have 1/3 pyrimidine that we can have, but not in DNA. Remember this guy here? We have something called RNA, and we're going to get into distinguishing the difference between DNA and RNA and where it plays a big role in your body. But the single ring structure, uracil, is only found in RNA. And it replaces thymine, if you recall from my other note. And we'll come back to this. So RNA only. Okay, so the four big ones that you're going to find in DNA, adenine and guanine, they're called purines. They're double ring structured. Cytosine and thymine, they're called pyrimidines, and they're single ring structures. It's important because it determines how the rungs of the ladder are going to go together, and that's what I'm going to show you next. Okay, so just to recap, here's sort of a summary. The sugar phosphate backbone makes up the sides of the ladder. The rungs are made of 4 nitrogenous bases. The ladder itself is made of 2 strands of DNA, which is why it's called a double helix. Now, the strands of each side of the ladder run anti-parallel. So this is an important term to make as well. And we're going to look at some examples of this. Anti-parallel means that, and let's maybe add a little note in here. Anti-parallel means that all it means is the sides of the ladder run in opposite directions. That's what anti-parallel means. Anti-parallel just means this. The sides of the ladder run in opposite directions to each other. Okay, so each strand is attached by hydrogen bonds between the nitrogen base pairs. So adenine, which is a purine, pairs with thymine. This is always who it pairs with. And G always pairs with C, guanine with cytosine. Now, the one thing you're always going to notice here, and I've made a big note down at the bottom, that a purine will always pair with a pyrimidine. And that's due to their chemical makeup and the number of hydrogen bonds that are going to form and hold together the strongest is always when you have a purine with a pyrimidine or a single ring structure with a double ring structure. So those are the rules that tell you which one pairs with each other and the number of hydrogen bonds that exist between each of these pairs. So this is something to remember as well.